So basically, this graphic summarizes some of key components in vaccines, as well as clarifying their purpose and safety in the concentration present. You may have a read and look this graphic in details on the description of each of the component, and it also provide some answer on what the anti-vax always claim on the side effect of a vaccine, and how these ingredients that are used in a concentration that will not cause any unwanted side effects. Next is on vaccine adjuvant. Basically, an adjuvant is an ingredient used in some vaccines to create a stronger immune response. In simple words, adjuvant help vaccines to work better. Some vaccines that are made from a weakened or killed microorganism contain naturally occurring adjuvants and help the body to produce a strong protective immune response. However, most of the vaccines or the modern vaccines being developed now include just a small components of the microorganisms, be it just a protein, DNA or RNA based vaccines, which is fully immunogenic on its own. Thus, this type of vaccines required administration with adjuvant to work effectively. So in this advanced era of technology, the search for new vaccines quite challenging, and sometimes you can't simply use the traditional approach where you can just weaken or inactivated it. Sometimes you need to have a more fancy approach which will generate a better safety profile, but you also need to search for an effective adjuvant to be used in concordance with these vaccines to generate the immune response that can confer protections. So basically, a key adjuvant function is to overcome the poor immunogenicity of subunit vaccines by improving pathogen recognition and eliciting a response similar to the innate immune response. And recently, nanoparticles technology are of great interest to be utilized as vaccine adjuvants. So nanoparticles can be made from various types of biomaterials, like from the virus-like particles, from the liposomes, where all these can form the nanoparticles, and the antigen can be entrapped inside these nanoparticles, thus provide protection to the vaccine antigens. It can be made from a polymer and also non-gradable nanoparticles. Why nanoparticles? Because nanoparticles resemble a similar size dimension to the virus or to the pathogen, so that it can be highly recognized by the body, by the immune system, so that it can be acquired and activate the necessary immune cells like the antigen presenting cell, so that our body system can recognize this and further translate the encapsulated antigens and to produce the necessary immune response against these particular antigens with the help of the nanoparticles based vaccine delivery platform. And this is the vaccine component of the Pfizer BioNTech COVID-19 vaccines, where the active ingredient is the mRNA strand that encode the S protein of COVID-19, and the lipids component here is the solid lipid nanoparticles that's used to produce the solid lipid nanoparticles, which act as the adjuvant to boost the immune response of the encapsulated mRNA antigen, while also to protect it from being degraded. So various types of salts are used that act as stabilizer. Then we have sucrose, which is a type of sugar which act as cryoprotectant. Why? Since the mRNA need to be stored in ultra cold conditions, we need the cryoprotectants to prevent damage to the cells during freezing. Alright, so basically, despite the success of current vaccine, there is a clear need for the development of vaccine against a number of infectious diseases, for which vaccines are not yet available or are not sufficient. For example, like HIV, malaria, TB, and also a universal influenza vaccine. But interestingly, vaccine can also be developed against cancer. If we used to know that vaccines is always to act as a prophylactic, which is for prevention, in this new era with the discovery of the DNA or mRNA vaccine, it's now possible to have a therapeutic cancer vaccine, which we can use vaccine as a therapeutic approach in order to eradicate cancer cells. And basically, recombinant vaccines, when combined with new stabilizer and adjuvant technologies, will deliver enhanced vaccine safety, immunogenicity, and stability. After all, what we want to develop is a safe and highly potent vaccines which can confer adequate protections against the targeted pathogen. With that, thank you very much. So basically, it's really important for you to be able to distinguish between the meaning or between the concept of DNA or RNA based vaccine in comparison to a protein based recombinant vaccine. And for exam, you need to be able to really understand on how the DNA recombinant technology works. So for example, if the exam question asks you to discuss on how you can produce DNA or RNA or a protein, you should know what the difference between these subunit vaccines and what are the difference in terms of their productions. Right? I wish you all the best. Thank you once again.